Okay, the first technology we're going to cover is recombinant DNA. So recombinant DNA refers to the joining of DNA molecules, and it's produced by artificially joining DNA from different biological sources that are typically not found together in nature. And then we typically try to incorporate this DNA into clones, which are recovered copies of recombinant DNA molecule. So recombinant DNA technology is used to isolate sequences, replicate sequences, and often analyze genes. And so the first thing that you need to be able to do is actually isolate DNA from an organism. So maybe you want to study flies, maybe you want to study bacteria, maybe you want to study mice, maybe you want to study some sort of environmental organism that's important for some remediation project. Maybe you want to study something that is involved in ecology, very important for ecology. So almost always what you want, if you're a geneticist or if you're a molecular biologist, almost always what you want to do is study some sort of form of their DNA. And that's either to study, and there's all sorts of purposes for this. And so the first thing that you typically want to do would be to isolate DNA from that organism. So if I give you a fly, if I give you bacteria, and you know, a bacteria and a fly are built from many different components. So there's cell wall components, there's proteins within these, there's cellular debris, there's what makes the cell wall, there's membranes, there's lipids, there's all sorts of things that are within an organism. And so if we want to purify out all of those other things and only be left with DNA, DNA molecules from that organism, how do we do that? And so it's actually very straightforward. So, back in the 1950s, researchers figured out ways to isolate DNA from other biological molecules. And so there's a couple different ways that are important. So one thing you can do is you can add material that adds something that will destroy something that you don't like. So this is an example of this. So if you add detergents, detergents are soaps. The way detergents work is that they break up lipids, they break up fatty acids. So anything that feels greasy that you can't get off your hand, is water insoluble. And so detergents just simply make the lipids soluble, so you can remove them and wash these things off of your hands, for example. But if you apply it to a biological sample, such as like ground up flies, that detergent will break up the fatty acids that make up the cellular membranes. So that can get rid of the large pieces of cellular membrane that you would normally find. You can also, so that's something that just kind of degrades it. You can also add something like an RNase. So an RNase is an example of an enzyme that degrades RNA. So if you wanted to get rid of the RNA from the sample, you would just add this protein and then it would degrade it. There's other ways that you can remove a biological material without degrading it. So very often there's kind of multiple ways to do things. So an example of this is salts. So salts are often used in order to precipitate proteins. And so precipitation means that you take something that is soluble and you make it insoluble. And so once that happens, then you can simply use a centrifuge to spin down and separate soluble from non-soluble material. And so if you add salt, the proteins precipitate, but the DNA remains in the supernatant. And then if you spin it down, you spin down all of those proteins and you can separate it simply by just removing the liquid from the tube and throwing out that. And then DNA you can also precipitate. So if you add ethanol, which is an alcohol, this causes DNA to become insoluble. And so now the DNA can be removed by simply spinning down in a centrifuge into a pellet and a supernatant. So, you can do this using chemicals, or these days you can actually even just order kits that kind of do all this stuff for you. But the end result is that you can actually isolate DNA from a molecule, from an organism. So from a bacteria, you might isolate plasmids. From organisms, typically genomic DNA. And so what do you do with that DNA? So one thing you might want to do with that DNA is to cut it at very defined sequences. And so you start out with something that is a very large piece of DNA, so it's genomic DNA, and it could be 20 kilobases, 50 kilobases long. And maybe one of the things you want to do, and hopefully as we go into this a little bit more, it'll become clearer why, is actually break the DNA at defined sequences. So is there a way to break the DNA at particular sites? And the answer is yes, and that is due to the isolation and identification of what's called restriction enzymes. So restriction enzymes, these are identified in bacteria as a defense mechanism against bacteriophages. These are enzymes that cut DNA. So if you put double-stranded DNA in a tube, and then you added one of these enzymes, they would be able to cut the DNA into pieces, but only at specific sites, which are called recognition sequences. So these enzymes, if they see a particular set of nucleotides in a certain order, they can bind that double-stranded DNA and actually cleave the DNA. And this produces what's called restriction fragments. So these are just fragments of DNA. So the enzyme cleaves both strands of DNA, and that is because it actually recognizes the palindromic sequence. And palindromic sequences are nucleotide sequences that read the same in both strands. So an example of a site that might be recognized by a restriction enzyme is AACGTT. And so if it recognizes that, in this strand, it can actually break the DNA. But you want to not only break the DNA at one strand, you want to break the DNA at the other strand. And so if you use a palindromic site, this is something that is the same if you read it in the opposite strand. So, under here I've just written out the reverse complementary sequence, so T and A, GC, CG, AT, AT. So you notice if you have a palindromic sequence, this sequence, 5 prime, read from the 5 prime direction, is A, C, G, T, T. And then if we look at the opposite strand, starting from the 5 prime direction, because DNA binds is anti-parallel, we have the exact same sequence, A, C, G, T, T. So any restriction enzyme that can recognize a palindrome will naturally be able to cut both strands of the DNA. And so here's just a couple examples of restriction enzymes. These are the names, so HIN3, BAMH1, ALU1, SAW3A1, and these are the recognition sequences. So 2 of these recognize 6 base pair sequences, so AACGTT, and you'll notice they're palindromic, AAGCTT, and you'll notice it's palindromic, AAGCT2. BAMH1 recognizes the sequence GGATCC, GGATCC. Some restriction enzymes only require 4 base pairs to recognize it, AGCT, again it's palindromic, and then SAW3A1 as well. So the enzyme, nowadays there's hundreds of these enzymes that you can order, and they all recognize their own individual recognition sequence. Further, they also cut the exact the exact location where they cut the DNA sequence is different. So for HIN3, it cuts between the first nucleotide and the second nucleotide. And so this is where that cut occurs, in between the A and the A. This is where the cut would be on the other strand between the A and the A. And so when we actually do this cleavage, what we have is an overhang. So this is called an overhang because of the fact that they cut at not exactly precisely the same location on the different strands. So not only have we cut the DNA, but we've also left these sequences with overhangs or sticky ends that could potentially be used to re-anneal with other DNA sequences. So BAMH1 also cuts in between the first and the second nucleotide, and so the resulting sequence would be as so, and so you can see that there's a GATC overhang, and then here is a CTAG overhang right here. ALU1 cuts in the middle, and so because it cuts in the middle, there actually aren't any sort of cohesive ends, so this is called a blunt cutter because the DNA that's produced is blunt. And then you have an example of a 4 cutter that cuts right at the beginning of the recognition site and again make cohesive ends. So these molecules have been separated, but now they can actually come back together through hydrogen bonds in order to form, in order to come back together. And so these are called cohesive ends because they're compatible with each other. So if I had a piece of DNA cut with HIN3, and let's say I cut it into 3 pieces, so it'll be easier if I do it down here. So, let's say we have a piece of DNA and it has 2 HIN3 sites. Okay, so this start from the beginning. All right. So we have a piece of DNA, and it has 3 HIN3 sites. What comes out of this is going to be something like this. And we're just going to separate them by color just to make it clear which one is which. So we just cut this into 3 pieces. So each of these 3 pieces have an overhang. So these overhangs can actually re-come together and actually form hydrogen bonds. So these are still 2 separate strands, so they're very easy to separate back apart because there's no covalent bonds in the single strands, but you can actually bring these back together. And what is useful from a sense of creating new combinations is that one of the things we can do, one of the possible ways these can come back together is in an order that is different. So what do I mean by that? I mean that the first and the third piece can now come together because the ends of these are coherent with each other, are cohesive with each other. And so this allows us to start to mix and match DNA in ways that hasn't been done before. So this would be an example of taking a piece of DNA and removing a large segment of the DNA and creating this structure that is a chimera of the first and the third. And we'll get into a second how you make this change permanent. But you can also not only do this with HIN3, so if we cut with HIN3, but you'll also notice that some of these enzymes are compatible with each other. So they produce the exact same overhang. So BAMH1 and SAW3A1, they have a somewhat similar recognition site, where SAW3A1 recognizes a GATC, BAMH1 recognizes a GGATCC, but they both produce the exact same overhang. And so you can actually take DNA that has been cut with BAMH1 and it will form the same cohesive ends as DNA that's been cut with SAW 3A1. So this is another technique that people use in order to form new combinations of DNA. So once this happens, so this is again an example of how we can form a chimera. So we take DNA, so let's say this is fly DNA right here. DNA we got from a fly, and this is bacteria DNA right here. So these are two sources of DNA with completely different sequences. And now let's say we take both of these and cut with this enzyme EcoR1, and so we are going to be left with DNA that has this overhang right here. Okay? And we're going to be left with DNA that has this overhang right here from the bacteria sample. So if we mix these together, one of the chimeras that can be formed is between this fly DNA and bacteria DNA, as long as the overhangs are compatible. So this is called annealing, and so the initial thing that happens is the DNA strands anneal together, and they're held together by hydrogen bonds. They're not covalently bonded because there's these small nicks that would still remain. However, this is where another important member of the toolkit comes into play, which is DNA ligase. So if we take this enzyme, so we recognize this enzyme from DNA replication, so this is used in order to join DNA sequences, producing Okazawa fragments, for example. And so we can use this in order to make this combination permanent. So the 3' DNA ligase seals the nick. So now, instead of it being a gap and it having a nick there, now we've made a fully covalently bond set of strands on both the Watson and the Crick strand. All right, so what do we do with this? So now we have the ability to form chimeric sequences, and very often the chimeric sequence that we try to create is a chimeric sequence with a vector. So vectors are one source of DNA that we can use, and these are typically derived from bacterial plasmids. So we've already seen some examples of bacterial plasmids in the F factor in Chapter 6. They replicate independently in bacterial cells. There's lots of plasmids per bacterial cell, and this is just some EM pictures of kind of what they look like. So now, over time, we have engineered plasmids to, instead of being the wild form of the plasmid, we have engineered them to be useful for our purposes as DNA engineers. So what are the things that we might add? So one of the things we might add are restriction sites. So typically they'll have multiple cloning sites. So this is, this might be about 100 base pairs long, but this small piece of DNA has all sorts of different recognition sites for different restriction enzymes. So HIN3 we saw before, SPH1, PSD1, these are all different restriction enzyme sites that will be recognized in cuts. So we can cut this DNA at all sorts of different places. So these plasmids have an origin of replication, so this allows it to replicate in bacterial cells. Another thing that's important to have is a marker gene. And so the marker gene that we're going to use is an antibiotic-resistant gene. So ampicillin is an antibiotic that normally kills bacteria. And so if they have this gene, the bacteria cells won't be killed by the ampicillin antibiotic. And so why is this important? So this is called a marker gene because it allows us to determine whether or not the bacteria is carrying the plasmid. So if the bacteria does not have the plasmid, it's susceptible to ampicillin and will die. If it does have the plasmid, then it's going to live. And then often they have DNA sequencing primer sites, so these are just convenient places to start where DNA sequencing can occur, so we'll get into that later in the chapter. And typically you might want to sequence from both sides, so there's sequencing sites that allow you to sequence in both directions. And then finally, another common technique is to use this gene called LAC-Z, which we'll get into in a second. And this just allows us to determine if something is cloned in here. So we are trying to get DNA from another organism into this plasmid at this site right here by using these restriction enzymes. So how do we do this? So we have our host bacterial cell. It has the plasmid vector. And so we talked before that we have ways to isolate DNA from bacteria. And so one of the first things we do is we purify the plasmid out of the bacteria. Then we cut the plasmid with the restriction enzyme. And so the double-stranded circular plasmid is now a linear piece of DNA. So this linear piece, for example, would not be able to replicate in bacteria because bacteria need to replicate circular DNA. We also have our foreign DNA source. So let's again say this is flies. So we have DNA that we've isolated from fly. And so we cut the DNA with the exact same restriction enzyme. And so we break this genomic DNA into a large number of pieces, and then we mix and match kind of all of this with this, and we ligate them together. So all of these, this also could have made its way into the plasmid. This also could have made its way into the plasmid. It's just kind of a random process, of which one makes it into. But one of these will be able to form, to match up with these sticky ends, and if we add ligase, DNA ligase, now we've recreated a piece of circular DNA. So this piece of circular DNA now has extra material. We've made a chimera between the fly and the bacterial DNA. So now we can reintroduce this plasmid into bacteria host cells by transformation. So we take advantage of the fact that bacteria have the ability to take up DNA. And so if they're able to take up this entire circular plasmid, this plasmid will be able to replicate and grow. And so how do we know that this has happened? So one of the things we do is we plate, so just to make it clear, so now we are starting with bacteria that has a host chromosome but does not have the plasmid. And so we can try to transform that, so some of the cells will take it up, some of the cells will not. And so how do we know which cells have taken it up? So we plate them on media that contains antibiotics, and that's because we know that this vector has a marker gene that confers ampicillin resistance. And so this is the way we can kill the cells that don't have it. So what are the potential outcomes from this? So remember, so yeah, so in this case right here, we saw what happened when this piece of DNA incorporated into the plasmid. You could have also seen this piece of DNA incorporate into the plasmid. You could also see this piece of DNA incorporate to the plasmid, et cetera, et cetera. And then one final possibility is that instead of anything incorporating into the plasmid, you just reconnect these pieces together. And so then you'd be left with the original piece. And so this is why this figure right here shows, this original orange plasmid versus something that has incorporated new DNA. And so one possibility is that, you just recreated your same original plasmid. And so this is why we also include a ligase gene in the plasmids that are engineered, and that just allows us to tell if there's been foreign DNA that has actually been inserted into that multiple clonal sequence site. And so how does that work? So that works because ligase encodes a enzyme called beta-galactosidase, And a beta-galactosidase is something that cleaves sugar. So some sugars are dimers or are created from two different sugars. And what they do is they just cleave that sugar into the two components. So, in general, that does not seem very useful, but we have identified a very cool molecule called XGAL. And so XGAL is not a normal sugar that your body would use, but it's an analog of lactose, so it's a modified version of lactose, which is a substrate from beta-galactosidase. And so when XGAL is cleaved by the enzyme, it actually turns blue. So if this LAC-Z gene is functional, then it's cleaving the XGAL that's in the media and turning everything blue. So the bacteria cells that don't have this are going to be white. And so this can be useful in two ways. So if the bacteria, so first we know if the bacteria, so if they have the normal plasmid, they have an intact copy of the LAC-Z gene. And so if they have the vector, then these cells would be blue. But if we actually successfully incorporate foreign DNA into the multiple cloning site, we interrupt the LAC-Z gene. So the LAC-Z gene no longer is able to encode a functional protein. And so these cells, which we know have the vector because they're ampicillin resistant, so anything that takes up this will be AMP-R. These will be white because they no longer have the functional LAC-Z gene. This guy right here, these will be AMP resistant because they have the vector, but they will also be blue because they have the functional LAC-Z beta-galactosidase, which is cleaving the XGAL. So finally, I want to summarize a little bit about vector. So here we just kind of talked about generically a way to get DNA material into different vectors, but we haven't really talked about why you might do that. And so there's all sorts of different types of vectors that are useful for different purposes. So there's bacterial artificial chromosomes and yeast artificial chromosomes. So the first one comes from bacteria, the second one comes from yeast. And these are vectors that can actually clone in a large fragment of DNA. So we're talking about like 50K to 100 KB pieces of DNA. So these are actually very useful for the sequencing of the human genome. So the original way that the human genome was sequenced is that we've incorporated human genomic DNA, the entirety of the human genomic DNA, into these BACs or YACs. Both of these were actually used, and then the DNA sequencing occurred from there. And this was actually necessary in order to create a full version of all 23 chromosomes. You also might add them to expression vectors. So these vectors are designed to take in genes, and they're designed to ensure mRNA expression of cloned genes. So why would you want to do that? Maybe you want to study the protein that is actually produced from a gene. And these have also been very useful for isolating insulin. So like if you wanted to create like a foreign, we'll get into this in a bit, but in the next chapter, but if you want to create purified insulin protein, one way to do that is that you just incorporate the insulin gene into one of these expression vectors. And then gene editing vectors, so we use these as a repair or recombination template to edit a host genome. And there's way more types of vectors that are used. This is just to give you a kind of sense, though, that the toolkit of plasmids is very large, and these plasmids incorporate additional sequence and additional markers and additional things that actually help you for whatever purpose that you might have.